Again, many thanks, everyone, for your talk so far. I'm really finding this workshop so stimulating. I've learned a lot, and it's helped me to look at my own research materials in a new way, because data linkage on the whole isn't a topic that's been front and center for me. So let me begin with an image that I was introduced to last week in Richard and Mario's talk. It's an image, as they put it in their paper, of possible new data linkages made possible by integration of new data. And what fascinates me about this is the ideal it presents of kind of frictionless movement across time of data. It's likewise an image that one gets from the phrase historical data mining that they use. A sense of data is kind of like oil. It's out there. And if you can only tap it, it will flow in useful ways. Now, I don't mean to suggest that for Richard and Mario, there are no impediments to be faced in realizing this vision. But on the whole, the sense one got was that these will be chiefly infrastructural. Data locked away in journals that no one's digitized, or in filing cabinets, or on floppy disks in a language that no one has machines that can run anymore, that kind of a thing. And undoubtedly, those will be problems. But my concern today will be on a different class of potential impediments, what I'm going to call intellectual impediments. And my particular focus, there may be other kinds, but my particular focus is going to be on a canonical problem in the history and philosophy of science, the problem of data that are theory-laden in the phrase. So here's my argument in brief. The basic thought is that the categories used in classifying observations and the choices made about which observations to file under which categories can reflect background theory. What I'm saying here has already been anticipated in a wonderful exchange just now between Helen Curry and Richard Osler, and it was cheered on by, I think it was Richard Davey. So I hope that this will seem the more apropos. So that's the basic thought, and I want to explore that thought with two stories, what I'm calling parables, about the exemplary Mendelian organism, the garden pea. And I'm calling them parables because in an old-fashioned way, I'm going to draw morals. And here are my morals in brief. The moral from my first pea parable is pessimistic. And it's that the problem of data theory-ladenness, close kin to the relationship of anachronism that Helen was talking about, needs to be taken seriously. That's the moral of the first parable. The second pea parable, however, is going to be optimistic. And the thought here is that one way to overcome the problem of data theory-ladenness is to retain access to the seeds of the plants featuring in historic data. So here my talk will intersect with another talk from last week, Helen's own talk about seed banks. So that's where we're going. Let me introduce my pessimistic parable first of all, the one to do with the theory-ladenness of data. Many of you will be familiar with this theme, but for those who aren't, the nub of the claim here is that it's naive to think of observation as just kind of sense data hitting our sensorium, because there can be theory or background knowledge conditioning what we observe. What we expect to observe can condition what we observe and what we report. So if I lead you to expect that you're going to be looking at ducks, you may see ducks. If I lead you to expect you're going to see rabbits, you may see rabbits. What's that got to do with Gregor Mendel? Well, on the whole, when one is introduced at every level of education to Mendel and his experiments with hybrid peas, you're not invited to think about the theory-ladenness of observation. Quite the contrary. What's emphasized is just how careful Mendel was in every way. He was a brilliant observer, and that's why Mendel was able to see the patterns that no one else saw. Here we're looking at the famous 3-to-1 ratio of dominant to recessive traits in the pea for seed color, right? Yellowness and greenness, yellowness dominant, to green in ways that Mendel not only described, but explained in such a persuasive way, right, by his postulation of factors for yellowness and greenness. And as you go on in your education, you are introduced to more generalized Punnett squares, that theme of the simplicity of the patterns awaiting in nature for those who are careful to take the time as Mendel did is, again, one that's emphasized. And I think what's not emphasized often enough, and I've been banging the drum about this lately, is that Punnett squares should have a big sign hanging over them that say, all else being equal, these are the patterns that you can get. That is to say, if deliberately you dial down all of the natural variability internally and externally, right? Expunge, as Mendel did, through vigorous purification, everything that might be getting in the way, genetically and otherwise, of the patterns that you seek, rigorously ensuring that you keep your environments uniform so that the only difference is a genetic difference at one locus on one chromosome. Yeah, at that point, you may get the pretty patterns that you learn about in the textbooks. But otherwise, well, all bets are off. And that's true for peas as well. We're looking here at a photographic plate in an article published in the journal Biometrica in 1902. If you just have a look at the top row, what you see are, in box number 1, pea seeds that are green. In box number 6, peas that are yellow. And between 1 and 6, peas that fall within a color spectrum between green and yellow. Other people's peas didn't look like Gregor Mendel's categorical green or yellow. And indeed, if you weren't expecting to see only green or yellow, you aren't tempted to describe them that way. This has been shown for students in our own day. If you give them only 2 categories in which to bin the pea seeds they're given, they put them into green or yellow. But if you give them 3, right, green, yellow, and ambiguous, they'll use 3. That photograph and that insight is due to this man, W.F.R. Weldon. He was, in the early 20th century, the Linacre Professor of Zoology at Oxford. This article, as I say, was published in 1902. That's just 2 years after the famous rediscovery of Mendel's paper, the year in which Mendel's paper rather unexpectedly became a talking point in European botany. Weldon was fascinated, but he rather quickly persuaded himself that it was a mistake to make Mendel's paper the centerpiece of a new science of heredity. Right? And it was a mistake because what you gain in control in a Mendelian experiment, you lose in generality. You end up with a very misleading picture of how heredity works. It was a misleading picture in 1902 on Weldon's view because it was already so well understood that what capacities tissues express depend radically on what they're interacting with. And that would be just as true of chromosomal tissue as it was for all other tissues. Why on earth turn our backs on all of that knowledge in favor of a view of heredity, which encourages us to ignore all of the variability brought about by interactions with multiple causes, internal and external? And likewise, why work with these categories, these binary categories, greenness, yellowness, roundedness, wrinkledness, when absent the rigorous attempt to make them that way? That's not what we see. We should want, Weldon thought, a theory of heredity which deals with all of the data as we actually observe them, not with a fictionalized, idealized version of that. The same paper in which Weldon published that photograph is also the one where, as he pointed out for the first time, Mendel's data were pointed out as too good to be true. That's something we associate with Ronald Fisher, but it was Weldon who pointed it out. And for Weldon, though, it was never a matter of fraud, or not in a serious way. It was rather, again, to do with those categories. because Mendel worked with such gross categories, because his theory led him to bin whatever he saw as either green or yellow, inevitably he put the observations where it seemed as though the ratios that his theory predicted required them. So that's my pessimistic parable, right, with the Mendelian exemplary organism, the pea, as my example. Theory can color observations and as they're reported. And from 1900 forward, Mendelism, the rise of Mendelism, the central science behind what later is called genetics, has been such a pervasive feature of the reporting of observations about plants, but more widely, that I do think the potential problems of the theory leadiness of data needs to be taken seriously. And one upshot, going back to my opening image, for us is thinking about the standard of distinctiveness, uniformity, and stability, and the ways in which that standard and data collected around it is itself a legacy of that Mendelian emphasis on control. Let me go from downbeat to upbeat. My optimistic parable, staying with the pea. You see there on your left an ordinary pea plant, but you see on the right a deviant one, or in agricultural terminology, a rogue. In this case, it's called a rabbit-eared key because of the way the leaves look at the bottom. Weldon took rogues very seriously, plants that deviated from the advertised type. He thought that's just what you expect to find, right, in a natural world where there is an awful lot of variability to be excluded by people who want to exclude it. Bateson, William Bateson, you see there on your right, the Mendelian in chief, on the contrary, often declared and loudly that Mendelism had solved the rogue problem. All you need to do is make sure you're working from homozygotes and you will have no rogues. Nevertheless, Bateson, once Mendelism was secure as a science, began to take an interest in a problem that plainly Mendelism hadn't solved. And in the 1910s, working with Caroline Pellew at the John Innes Institute, they undertook a fascinating research project on rogue peas. They could never quite work out what was going on, but they took the problem seriously. And they took the problem seriously in part because it remained a problem even in Mendelian agriculture. We're looking here at Roland Diffin, the great Mendelian breeder of the era, right? the man whose reputation was built on having applied Mendelism to agriculture. This is a jokey image produced by his students in the early 1920s, right? No one's ever held a sheaf of wheat that big. In the image, it's called Yeoman 24. And that's a fascinating name because already by the early 1920s, it was well understood that Yeoman, the variety of wheat released in about 1916, had become problematic. It was rogue riddled. By the early 1920s, Biffen was saying that the sooner Yeoman, this Mendelian wheat, was removed from the market, the better. So the rogue problem, the problem of unwanted variability, remained a serious one, theoretically and practically. But to go back to the piece, or as I say, Bateson and Pellew never bottomed out what was going on behind the rogue phenotype. But the seeds that they collected remained available thanks to the Germplasm Resource Unit, as it's known now at the John Innes, and from time to time attracted the interests of other researchers right through to our day, a day in which, of course, we've had all kinds of expansions in knowledge of heredity, not least expansions in understanding of what's called epigenetics, right? The possibility that the differences that we see are due not to differences in the DNA sequence, but to the chemical overlay, not least DNA methylation patterns. And indeed, in just a few years ago, a group of Portuguese researchers re-examined the material that Bateson and Pellew had worked with and concluded that indeed the rogue pattern was due to methylation patterns. So let me draw to a close My two parables, again, the pessimistic one, the problem of data theory relatedness needs to be taken seriously, I think, and I think that the story of Mendelism is a helpful one for helping us do that. But there's also an optimistic message from my second parable, that one way to overcome the problem of data theory relatedness is to retain access to the seeds of the plants featuring in historic data. So am I saying that that's the antidote? Right? As long as you have access to the seeds themselves, all the problems of anachronism and theory relatedness go away? No, of course not. That would be terribly naive. Nevertheless, I think that there are grounds for optimism and even exciting activity. And I want to close here with a plug for Nalini Kochupillai. Her talk will be taking place next week. From Nalini, I learned that in Albert Howard's Agricultural Testament, recording observations that he'd made as a student of traditional Indian agriculture in the 1930s, he reported sugarcane yields higher than any yields then seen, then now seen. And I think it's a fascinating question to ask whether the seeds behind the sugarcane that Howard was observing, might those still be available? And might it be possible to grow them again under the soil and climatic conditions that obtained when Howard observed them? That combination of genes, genetic background, and environment. If those yields happen once, then they can potentially happen again, and that could be instructive. And I'll end there. Thank you very much.